Ever stop to think about how your body manages something as basic as moving potassium in and out of your cells?
It seems simple, right?
Yeah, you'd think so. But it's actually a really precise process, and it's going on all the time, makes your muscles work, your brain, fire signals. It's pretty crucial.
Absolutely. And today we are diving deep
Deep into potassium channels.
into potassium channels.
We've got some really interesting research on how they actually work, like at the atomic level.
We're going to be looking at the actual structures of these channels.
Our mission today is to figure out, how do these structures actually explain how potassium channels work so well? They're incredibly selective for potassium, and they can move it across cell membranes so fast.
Yeah, they're really efficient little machines. We'll be looking at how their physical design allows them to carry out those essential functions.
Okay, so let's get into it. All the potassium channels that we know about, I mean, whether they're in bacteria, plants, or in us, they all belong to the same protein family. And all of them share this tiny sequence of amino acids.
Right. It's like a universal key that unlocks potassium channel function across all domains of life.
Oh, wow, across everything.
Yeah, bacteria, archaea, eukaryotes.
So what does that tiny sequence actually do?
Well, it forms the core of a region called the selectivity filter.
The selectivity filter. Okay, so it's selective for potassium. What does that mean? What's it selecting potassium over?
So this filter is what gives the channel its amazing ability to let potassium ions through, but to pretty much block sodium ions completely,
Sodium ion.
even though sodium is actually a lot more abundant in our bodies.
Oh, interesting. So there's way more sodium around, but the channels are saying, nope, potassium only. So how much bigger is potassium than sodium?
Potassium ions are only slightly larger than sodium ions.
Yeah, way larger.
It's a tiny difference.
Sure.
We're talking less than the width of a hydrogen atom.
What? Less than a hydrogen atom. But these channels are over 1,000 times better at letting potassium through, and they do it incredibly fast.
That's right. It's almost as if the ions are just floating in water.
It doesn't make sense. How can such a tiny difference make such a big difference in selectivity?
Well, the answer lies in how elegant the design of the channel's pore is.
The pore.
Yeah, it's built from 4 identical subunits that are arranged in a circle.
Like a little tube.
Right. And that creates the central pathway for the ions to move through.
Okay, so 4 subunits making a tube.
Yeah. And this pathway has kind of inner and outer components. There's a special helix with these negatively charged ends pointing inwards.
Oh, okay. What do those do?
And importantly, right in the middle of the membrane, which is this oily environment, there's a water-filled cavity.
A water-filled cavity.
Yeah.
That sounds important.
It is. It helps to stabilize the potassium ions because normally they're surrounded by water, so it kind of helps them feel more comfortable as they enter the channel.
Okay, so like a little rest stop for the potassium ion.
Exactly.
Out of the oily membrane into this little pool of water.
Right. It helps them make the transition from the watery environment outside the cell into the oily environment of the membrane.
And then what happens?
That's where the magic happens. It happens at the selectivity filter, that signature sequence we were talking about,
Okay.
which is located on the extracellular side of the pore, so the outside of the cell.
Right.
And it's made up of these layers of oxygen atoms. So as the potassium ion enters, it loses its water shell, right? And these oxygen atoms that are part of the channel protein, they kind of step in and act as these perfectly positioned little hands that hold the potassium ion in place.
So they mimic the water molecules that it would normally be surrounded by?
Exactly.
But why doesn't sodium get in? If it's just a tiny bit smaller, shouldn't it be able to sneak through?
Well, sodium is actually too small to interact in the right way with those oxygen atoms.
Huh.
It can't form enough stable bonds to make it worthwhile for it to enter the filter.
Okay.
It's kind of like trying to fit a key that's too small into a lock.
It just doesn't work.
It just won't work.
Huh. So interesting. And there's another interesting thing about the selectivity filter.
Yeah, so when potassium levels are low, the filter can actually kind of squeeze shut.
Oh, really?
Yeah.
Like it's saying, hey, there's not enough potassium out here, we're shutting down.
Exactly.
That's so cool. Okay, so we've got this super selective filter, but it's also super fast at moving potassium through. How does it manage both?
Well, that's where things get really clever. It's a 2-part trick.
Okay, lay on it.
There are usually multiple potassium ions inside the selectivity filter at the same time.
At the same time?
At the same time.
Okay.
And remember, potassium ions are positively charged, so they actually repel each other.
Ah, okay.
This repulsion helps to push the ions through the channel.
Like a little conga line of potassium ions all pushing each other forward.
Exactly.
Okay, so that's part 1 of the trick. What's part 2?
Part 2 is that the shape of the selectivity filter itself can actually change.
Hmm.
So when a second potassium ion binds, it causes a small structural change in the filter, and that makes it even better at letting potassium through. So the very act of binding actually speeds things up.
Wow, that is so clever.
Yeah.
It's using the binding energy to actually make itself more efficient. Okay, now these channels don't just stay open all the time, do they?
No, they don't.
They need to be controlled, and that's where gating comes in.
Right.
So how does that work?
Well, by looking at the structures of potassium channels in both their closed and open states, we've learned that the main movement is in the inner parts of the channel.
Okay.
So when the channel is closed, these inner helices are straight and they kind of bundle together, blocking the
Like
pore.
closing a gate or like a drawbridge going up.
Exactly. But when the channel opens, these helices bend at a specific point, almost like a hinge, and then they move apart. And that opens the pathway for the ions.
So something triggers this hinge to bend.
Right. And what's really cool is that there's this one particular amino acid, glycine, that's often found right at that hinge point.
In lots of different potassium channels.
Yeah, across many different types. So that suggests that this is a common mechanism for how they open and close.
It's like the universal hinge for potassium channels.
Exactly.
Okay, so that's gating in general, but what about channels that respond to changes in voltage?
Right. So those are called voltage-gated channels, and they're super important for nerve impulses.
And how do they sense voltage? They must have something charged that moves, right?
That's right, they have what are called gating charges.
Gating charges, okay.
And in voltage-gated potassium channels, you actually have a significant amount of positive charge concentrated in this one region called the voltage sensor paddle.
The voltage sensor paddle.
Yeah, it's kind of like this little arm with these positively charged amino acids.
Okay.
And the theory is that when the voltage across the membrane changes, these paddles can move within the membrane.
So they're actually physically moving?
Yeah, near the protein-lipid interface,
Wow.
which then triggers the channel to open or close.
And is there any evidence that these paddles actually move?
Yeah, experiments have actually shown that these paddles do move back and forth across the membrane, depending on the voltage.
That's wild. Okay, so to bring it all together, the atomic structures of these potassium channels have revealed how they do this incredible job of being selective for potassium and moving it so fast.
It's remarkable.
We've got this amazingly designed selectivity filter, the multiple ions helping to push each other through, and these dynamic movements of protein structures controlling the opening and closing.
And all these processes happening at such a tiny scale are essential for life as we know it. It really is remarkable.
It really makes you think, now that we have this detailed view of how these tiny protein machines actually work, what does this mean for the future of medicine?
Yeah.
Could we design drugs that target these channels when they aren't working properly? I mean, think about it. Manipulating these mechanisms to improve our health, it's pretty mind-blowing.
It really is.
And that's it for today's deep dive.
Thanks for listening.
See you next time.